We're going to be talking about coelomate animals for Unit 3, so I wanted to just quickly go through the two developmental pathways that coelomate animals are known to develop. So those two pathways are basically the pathway that leads to protostomes and the pathway that leads to deuterostomes. So protostome literally just means mouth first development. Deuterostome means mouth second development. This is a very simple phylogenetic tree for animals. And if you notice, go to the bottom where you've got the ancestral protist. As we come up from there, we've got the asymmetrical animals, just as blobs there. You've got the radial animals, which are cnidarians and ctenophores here. And then if we take the bilaterally symmetrical branch, it comes up and we've got two branches again. The left-hand branch says no body cavity, which isn't actually quite true here because some of those animals that are even pictured there, like the nematodes, they have a body cavity, but they're pseudocoelomates. Animals with a true body cavity are the right branch. And if we come up there, there's another branching point where you go to either protostome development, which is mouth first, or deuterostome development, which is mouth second. So that's where we are in the animal kingdom. We're just on the right-hand side of this phylogenetic tree. Now these two different modes of development are really named after how the animal develops early on. So if you look at the two columns of drawings, you've got the blastula phase, and of course if you're on the left-hand column is the protostomes and the right-hand column is the deuterostomes. And of course it looks the same. They're different drawings, but you can't tell them apart really. They're the same. And then you get to the gastrulation phase where instead of just a hollow ball of cells, a pocket forms inside. And that pocket is going to become the gut of the animal. And when that pocket reaches the cells in the back of the ball of cells, that's when it forms the tube, and that's going to happen in either case. It doesn't matter whether you're talking about the right hand column or the left hand column. Now once it does that, now you've got the basic structure of a eucoelomate animal. You've got a mouth at one end, a digestive tract going through the middle, and then an anus at the other end. Now and again, if you look at the bottom two pictures, they look the same. The difference between them is not what they look like. It's just what does this first opening, what is this going to become in the adult animal? And that one is going to become a different structure than this will. In a mouth first animal, that first opening that forms becomes the mouth. The second opening that forms becomes the anus. In a mouth second animal, that first opening that forms becomes the anus, and the mouth is the second opening that forms. So these are just two different developmental pathways that really get you to the same end. But they go along with a whole bunch of differences that uh, that have to do with a bunch of other things as well. The at the moment of fertilization, that basic symmetry and body plan is determined, and it varies from one animal to another. Frogs have been studied a lot because frogs have external fertilization, and they've got an egg that's clear, so you can watch development really easily. And in frogs, the point where the sperm fuses with the membrane of the egg, well, that point always is the basis of the bilateral symmetry, but what that point means varies from one species to another. In frogs, the point where the sperm fuses with the egg becomes the ventral side. Now, once the egg is fertilized and you start getting those early cell divisions, again, there are differences between the way protostomes work and deuterostomes work, and you can easily see these differences in a microscope. So with protostomes, if you look at the cartoon here, so we've got division starting now with 2 cells, 4 cells. Now when we go from 4 cells to 8 cells, we have this pattern. You're looking down at it from the top, but you notice that the 4 cells on the top are offset. Each cell on the top is basically sitting on top of 2 cells underneath. That's called spiral cleavage. Now in deuterostomes, the early stages you couldn't tell the difference. I mean, here we go. We've got two cells, 4 cells, but when you go to 8 cells, the pattern is very different. Now you wind up with an 8-cell stage that looks like this. So you notice that each cell is basically just on top of one cell below. It's not kind of rotated a partial turn. And we've already talked about gastrulation, that difference, whether it's mouth first or mouth second. Okay, now the actual literal formation of the coelom can happen in one of 2 ways. In protostomes, the mesodermal cells actually split and expand to form the body cavity or the coelom. And if you look at the deuterostome side, the coelom starts forming as an outpocketing of the gut tube. Now, if you look at this drawing and compare it to this one, they look really different. But if you look at the end result, look at this drawing and compare it to this one, they're not really different. So, the end result is the same, but there are 2 different developmental pathways that occur that get you to that end result. Now, another difference between protostomes and deuterostomes has to do with basically the way that those cells in the very early stages map to the body of the adult animal at the end of development. So if you take a look at the protostome side here, we've got an embryo with 4 cells. If you go in a microscope and remove one of those cells, now we've only got 3 cells left. What you have effectively done to that developing animal is that you've removed a quarter of its body. That one cell you removed was ultimately going to develop into approximately a quarter of the animal's body. So development just stops. It doesn't work. The embryo dies at that point. Now, if you go into so they call that a mosaic embryo. So that's how it works with protostomes. If you go into look at the same stage in a deuterostome, so here we are at 4-cell stage, you go in and pull out one of those cells. So again, we've got 3 cells left. I don't know why they're always showing the deuterostome embryos kind of in the side view, and it seems like they're always kind of showing a top view for the protostome embryos. But anyway, if you do that, it doesn't mess up development at all. Everything still develops normally. They call this a regulative embryo. So in other words, this embryo that has lost a cell has a mechanism in place that it recognizes that cell is lost, and it just replaces it. It doesn't screw up development. In a mosaic embryo, there is a mapping between each of those cells in the very early stages and major parts of the body. You don't really have that with a regulative embryo. All right, and here are just some of the groups. You've already talked a lot about protostomes with Dr. Baker. We're really going to be looking at the 2 major groups of deuterostomes, the echinoderms and the chordates.